Good day, I'm Dr. T and welcome to my office. So today I'd like to talk about antibodies. Antibodies are going to be specialized proteins that bind to targets. These targets are called antigens, and then the immune system can deal with those targets as best it sees fit. These are natively a defensive system for organisms. So the antibody is going to be a Y-shaped protein, and its creation is a rather interesting one. The cells that manufacture them are going to take the genetic material for the antibodies and delete most of it. Kind of like a meat and two sides one. Essentially, they're going to be picking at random certain parts of the antibodies. You know, one of these, two of those, that kind of stuff. And by doing that, the antibody is going to have certain regions of which that are effectively randomly assembled. Specifically, we want to look at the ends of the Y, so the tips of the antibody, not the inside part of the Y, where some animations put the binding, but it's actually on the tips of the Y. Because these are random, they're going to have random intermolecular forces, so they can bind to things kind of weirdly. Now, in the bone marrow, the antibodies and the cells that manufacture them are going to be tested. If they bind to stuff, they're probably binding to host proteins, and that's not good. Those are hopefully discarded. If they're not, then this leads to autoimmune diseases. Now, once they mature out of the bone marrow and are floating around the bloodstream, if they float around, don't bump into anything for a prolonged period of time, the cell is simply discarded. It's not needed. However, if the antibodies bind to something of interest, aka their target antigen, then, well, they needed to bind to that, and you know what? You probably need more, so the cells start replicating, and you start building up a quantity of those antibodies. This is how you fight off an infection and achieve immunity to the infection. The presence of antibodies, once developed, can last quite a while within an individual, however, not necessarily indefinitely, with some antibodies for pathogens persisting for the rest of the individual's life, with others lasting mere months. This is why shingles is a problem. Chickenpox, which is a fairly common virus, once it gets into an individual after the initial infection, it lays dormant within nerve cells. And as one's immune system loses the antibodies to fight back, the chickenpox virus can reemerge and give you another round of it, which we call shingles. This is why a vaccine, if you've had chickenpox towards shingles, is recommended once you get a bit older. Additionally, other pathogens like the good old flu will change the parts of the pathogens that are the actual antigens and randomize those up a bit, so now all of a sudden it has this fresh new take and your original antibodies may not work particularly well against it. Sometimes you'll get a little bit of a leg up where they'll kind of bind, but you need a little bit more adaptation. But in other cases, it looks like a new virus and a new target, which is why, flu shots every year. Not because you're losing immunity per se, but because the flu has changed. Okay, so you've got your antibodies. Now, obviously they're a defensive mechanism for you, incredibly important for that. What else can we do with them? Say a little bit more artificial or synthetic approach. Well, we can take the antibodies from an individual who has been infected with some disease and inject them into another individual who is currently fighting said disease. This is going to allow for kind of a jump-started immune response, specifically in cases where, say, you're building up antibodies, but you're going to be dead before you fight off the disease. So doing it this way, you've got a much better shot of fighting off the disease. We can see this with some of the discussions of recovered COVID-19 patients using their serum to treat currently seriously ill patients. Now, a few more approaches. One of the classic ones is going to be using the antibodies as a detector. So we can take antibodies, and these don't have to be from humans, and quite often they're from, rabbits, horses, whatnot, good old mice. You take an antibody to a certain antigen, and this is a technique known as ELISA, or enzyme-linked immunoabsorbent assay. So you're going to have a sample that you want to know whether or not it has a certain analyte in it, a thing you want to study for. So you get antibodies that will target that analyte. So that analyte is now also called the antigen. So same thing, just kind of different reference. So you're going to have some antibodies. And in one form of ELISA, you're going to take a well plate. So this is just basically a plastic plate with lots of little dips in it to make little teeny tiny beakers. And then they're all kind of hooked together just to make life easier. So you take a well plate, and the insides of the wells, the little itty-bitty plastic beakers, are covered in antibodies for the thing you're trying to detect. So you put the solution that you think has this. This could be something like urine or blood or whatever. You put that in the well. If the analyte is in there, the thing you're trying to detect, because it's the antigen to those antibodies, they will bind it and they will stick it fast. So at which point you now have a solution, you've got antibodies, and if the analyte was in there, if the thing of question was in there, it is bound to the antibodies. You now take that solution and throw it out. But the thing you're looking for is still in the well because it's stuck to the antibodies, and the antibodies have previously been bound to the plastic. Now, you put in another solution. Usually there's like a wash step or so, just to make sure everything's legit. You put another solution, and this will have more antibodies to that analyte. These will typically bind at a slightly different spot, just because it's getting crowded here. Now, these antibodies, directly or indirectly, have some form of reporting feature to them. This could be an enzyme on them that changes a substrate that you're going to add in later. Colors. This could be something that glows. In theory, it could be something that's radioactive, but that's not done because that's a lot of work that's unnecessary. Paperwork for radioactive stuff is just a nightmare, so we try and phase it out whenever possible. But there's going to have some way of reporting back. So you put the antibodies in with the reporters, and then you throw them out. Like, but why'd you do that? Well, if the analyte is in there, the analyte is bound to the first set of antibodies, and the second set of antibodies will bind to the analyte. Now, you then, after you've discarded the ones that didn't bind, you go and do whatever is needed to detect your antibodies. And sometimes there's like a two-step thing just to make life a little bit easier. Now, let's say you have a reporting enzyme. Horseradish peroxidase is popular. So the horseradish peroxidase is bound to the antibody that's bound to the analyte that's bound to the antibody that's bound to the plastic. If you have this, then you add its substrate and the material turns purple. But if the analyte wasn't there, then the horseradish peroxidase is bound to an antibody that got thrown out because it didn't get to stick to anything. Therefore, it won't turn purple. Thus, if it turns purple, you have your analyte. Can you think of a case where something changes colors to know if you have something? If you said pregnancy test, you're right. Pregnancy tests are a highly modified ELISA system. Now, we can actually modify this a little bit differently. We could take the well plate, we could take the analyte of interest, we can stick that into the well plate and glue it on directly. In this case, we're not going to be looking for the antigen, we're going to be looking for the antibody. And this has also received news. So you take the antigen and fix that to the plastic. You then take a sample of serum that you want to know whether or not it contains the antibody. You let that sit on there, and then you flush it off. Now, you have either antigen and nothing else, because nothing stuck, or if the antibody was present, then you have antigen that's bound to the plastic and an antibody that's bound to the antigen. You discard the serum, you wash it, and then you put in another antibody that has a reporter onto it. And so this way, if the second antibody is able to bind to the first antibody, and you can have general antibodies that will bind to just about any old antibody, because the tail of the Y, the droppy bit of the Y, the bottom part, doesn't change much. So that's fairly standard. So you can just have a, that Y sits in there, and then, hey, this antibody targets human antibodies, so you'll know if there's any human proteins or human antibodies present in your well. If there are, hey, guess what? You have the antibody that targets the antigen that's bound to the well plate. If you don't, then the reporting antibody will have been washed away. Now, ELISA is a wonderful technique. It's a little buggy to get started, but once you get it started, it's very easy to run, requires very standard equipment. It does have some reliability issues. So in certain high priority cases, where you have to make sure you get the right answer, it's usually recommended that the ELISA be run again, or I should say another technique used to confirm the ELISA. ELISA is a very easy, fast, cheap approach, so go for something a little bit more reliable, a little bit quicker. There was a bit of a controversy several years ago in Canada where a company was running drug tests, and it would take suspects, I think urine, I'm not for sure what biological fluid, they'd take their urine, run an ELISA. Now they were supposed to then take that urine if the ELISA was positive and then do HPLC or gas chromatography on that, depending on what they were looking for, which is a more laborious technique, but is much more reliable. They didn't, and thus they said quite a few people had been using drugs who were not. And yeah, that was quite a bit of an impact, because those saying, hey, you're using drugs, had major impacts on the individual's life, such as losing child custody. Lawsuits ensued once this was discovered. So that also has the moral of the story of make sure you understand the limitations of your assays. An imperfect assay can sometimes be very, very helpful to you, whether it be a screening assay to see which ones you just don't have to worry about, or in cases where, say, if you get a couple wrong, that's not a big deal. You need rough numbers. So how many people have a certain disease in the population? If you get one person off or not, as long as you're not using it diagnostically, not such a big deal, but you get a pretty good idea of what percentage of population has or has had said disease, which of course is in the news. It's just those people need to know that, hey, you may not be immune to this disease, still take care, but you probably did have it, kind of thing. So with all that said, when it comes to the current pandemic, because if this is in the future, when you're watching this, notice the upload date on this video. This was a fun year, wasn't it? But if you're watching this in the present, not long after I've recorded this, please make sure to get any medical advice concerning the current pandemic that's nice and updated from highly reputable medical sources. So with all that said, have a wonderful day and stay safe.